thank you so much. First of all, I would like to also thank Milita taking over on my spot. Dr. Arvin, thank you so much. And again, thank you for the opportunity. So as I was mentioning, as of today, I'm presenting the allogenic off-the-shelf CAR T-cell therapy. But it's more than that. It's an AI-driven end-to-end platforms that allow us to select targets. As you were mentioning before, targets in solid tumor are completely difficult. There is a heterogeneity. There is a lot of issues in the context of finding targets. So we developed platforms through an artificial intelligence and data mining system that truly defining an end-to-end product, which through Kiromic Biopharma we really developed I believe a first target AI-driven CAR T allogenic off the shelf. So with that, I would like to, of course, some forward-looking statements. This is the outline. I will walk you in the next 25 minutes, 30, a little bit of where we stand today in cancer immunotherapy, specifically also on the context of what is being developed and how the field is moving forward. Some of our development platforms, the gamma delta T cells, and in the end, some of the validation in animal models of our CAR AI technology using our development isoform Q that is broad expressed in different solid tumors. And starting with challenging, we were able to generate a platform thanks to the fact that the machine are truly learning. And we have this huge development in the area of computer science. They allow us truly to find and develop targets. I spend most of my early scientific life as a discovery targets on CT8 cancer testis antigens, and really difficult to find a target and validate and take into the bench from the bench to the clinic. I believe that this platform will help to have multiple shots on goal and allow us to eliminate false positive. This is a slide showing truly that machine learning are truly learning, and the data will make a difference, the training data, so they will make a good algorithms and a good platform for validation. So, and that is almost a joke showing this human that are learning from androids, but truly the androids is learning from proper data, in this case, the data sets. So this is just to showing what in the last, I would say, 150 years of development for the clinic to the biotech, and so for the discovery on the AI. So you can see here that there are 3 blocks from the clinical discovery, biotech in the middle, and then the computer science. So through the discovery of choline 1891 from the injection of inactivated bacteria, through the definitely Thomas Burnett and Jim Allison with the CTL4 discovery and the Nobel Prize of Immunotherapy, definitely in that period of time, there is a truly an immense era of an ecosystem that is developing through these new drugs, such as ipilimumab, and then starting with Cas9 in 2012. But what you can see here that truly is the neural network system on the end after the discovery of Cas9 and the neural network machinery, which is start developing after the single-cell RNA-seq next generation sequencing in 2010. You can see there's a huge amount of data that explodes and allow us to discover targets and allow to create a much better personalized medicine in the context of finding a platform like an off-the-shelf CAR T gamma delta in solid tumor. So this is, I think, is an important aspect and give truly a perspective of which what is the field is going and how we can see how these proteomics, genomics, and so forth can be helpful in the context of creating a solid neural network system or AI systems, and also data mining. So if you see here in the context, this is truly what AI immunotherapy is the next level, how we can interact into the context of developing all information. As you see, you are working with AI every day, from watching a movie with Netflix. They know exactly what you are consuming, what is your preference, what is your quality of life. And then on top of it, now the social media and drugs that can be customized based on your phenotype and characteristic of the data mining that has been occurred through your social and your digital personal life. So what truly is in now exploding is the hospitals, and so what is truly in the health system is being developing right now in genomics, transcriptomics, proteomics, metabolomics. So creating truly an omics concept, truly where Kiromics is trying to truly emphasize the omics and trying to digitize the information to allow a better response in the clinical arena to defining targets, a better robust immunotherapy approach in an off-the-shelf system. So that's what truly we see is our suite of neural network of software that allow us to define and determine and characterizing data from the public database and also from private databases, which are then comprised into a different softwares, a suite of softwares that are able to then defining targets for CAR, therefore on the surface, or for T-cell receptor, or for adoptive T-cell therapy, and therefore through MHC class 1 or either non-MHC class 1 restrictions, and through all the process of validations and the different characterization. I'm not going to go to the details, but there is a strong validation internally and a well lab and third party to allow us to define targets. So what truly what it means the discovery of targets. Definitely you will need 3-4 lives of each specialized scientist just to having a couple of shots to defining targets and discovering targets. What I believe that this new era of artificial intelligence and neural network system with the data mining can really compress time and the cost of taking drugs that are for targets therapeutic products into to the patients faster and avoiding all the issue that as a human we do have. And this is an example of what a suite of a neural network system that we have. We calculated with our computer scientists what it will take for defining a target, discovery, or defining a target for predictions for one indications. And the permutation was that you need basically for that particular results that our neural network system Diamond does. They can do it in 1 day before validation. We'll take 158 human employees to 1 year of working, so specialized scientists, and still there is a lot of issue on the validation. So what we have development on the target discovery that would be used into the context of gamma delta T cells is truly a 5-step system using a drug discovery through billions of data points through public and private, and defining targets. You can remember in the first slides, we was defining what they read is truly what you are, and what is your algorithm is truly what is the data training has, and how you select it in the beginning, the training, the permutation system of the algorithm of allow then having a much more robust software or suite of software. In this case, specifically on the step 1, we collect data privately, but also with private library, but also through the Cancer Genome Atlas and cancer immune data points and peptides database and public databases, and then we refine it also from clinical trials that we have analyzed After that, we have several different suite of softwares that can be AI algorithms or either data mining, big data software, such as Cancer Splice for selections on 3D visualization, iCloud and epitope selection for HLA class 1 and class 2. Then we have a different kind of system for binding selections. And then we select as one of the few, we have several different targets already in the pipeline that we are developing at the moment. The most advanced one is our ISO targets system, ISO mesothelin 2, that is being validated and creating a much more specific targeting for cancers, such as ovarian cancers. And then we have a very strong target validation well lab internally and externally on the step 4. And also with defining through the clinical trials, the AI modeling to continue improve the identification of patients, and therefore how they select or being selected into the trial, but also how being during the trial, how they provided the potential response of the treatment. And I think saving truly time, and time is this essence, which allow us to define more targets in the context of specifically on solid tumor. So this is an example of what we are seeing right now in what we have developed this particular system that defining these cancer splice targets on the surface, and we then generate all the validation that I will walk you through in the next step. So the first that you see is the diamond prediction systems. And then when we defining this particular target, in this case, this mesothelin isoform 2 system, we are able to do a prediction. So which are the indication of which particular cancer will express or highly express the particular targets that we are looking at. In this case, specific ovarian cancer was one of the super expressed on the surface. But there are also other indications that we are exploring, such as lung cancer, pancreatic cancer, stomach cancer. And then we are able to defining and creating the specific peptide that will be then incorporated into the customized regions, which allow us then defining our antibodies. So that is the mesothelin GPA anchor surface glycoprotein that we identify in the isoforms, then have the specific proprietary sequencing that we just create then a series of antibodies, which we will validate. You can see in the step number 2, we create antibodies and then we validate it. This is the results of some of the binding activity detected to the surface of for the mesothelin isoform 2 on the tumor surface, and we generate also different series of different hybridomas, monoclonal antibody with the canonic form, non-canonic form. We select the most highly is AC50 binding, allow us truly to have a high definitions of the binding activities on the surface. And you can see here, this is just a representative of the binding kinetics of it, and also the different, the two selected monoclonal antibody 11C11 and 1B6 in the context also to our cell lines that we generate our unique validation cell like NCI H2226 and also on primary tumors. And the next, of course, after these monoclonal antibodies, we provided the validation through proteomics. To the study on the proteomic of the ovarian tissue samples were cohort of 109 ovarian cancer patients, which 100% was serious adenoma carcinoma with the histological subtype of 81% of grade 3, 64% tumor stage and stage 3, and 15 on stage 4. So what we have done here, we have selected particular data analysis. There was a study published in JCI that we had were able to get these particular informations and data sets. And we're able to define in the expressions and the translation of this particular transcript of the mesothelioma and detect it to the tumor versus the normal samples to create a better specificity and expressions or validation of the expressions of our isoform tube. In this case, specifically, you can see here. So that creates a much more robust and much more specific therapy in the context of having a much more effective or targeted therapy. So with that in mind, we had passed through some of the validations that we have done with several different institutions that collaborated with us on the validation, from Baylor College of Medicine and University of Milan, and now we're working also with some specific in MD Anderson for the pancreatic cancer. What you can see here, we have a representative panel of tissue samples, clinical samples from patients, specifically for ovarian cancer. You can see where you can see normal ovarian tissue that is truly negative on your left, and high-grade serous carcinoma, truly you can see this brown area truly highly expressed. Then normal pancreatic tissue, you can see on your left on the panel, this is negative, truly expressed, and then on pancreatic, again, adenocarcinoma. And the normal meso-Lang mesothelioma or Lang versus the primary mesothelioma as a tumor, you can see there are spots of expressions in the selection of the tissue and the fields. So, and then we selected, of course, on very specifically on ovarian cancer, you can see on your left through the panel of our selected antibody 11C11, truly highly expressed in ovarian cancer. So that it's a study that is ongoing with cell lines, but also with primary tumors, as you can see, for different targets and different indications that was predicted by Diamond AI system. And so the prediction was given, and we knew that it was expressed in ovarian cancer, but truly others were still in the process to expand our database on the different solid tumor. But that is truly showing that there is a validation, not just on the proteomic aspect, but also on the selecting patients that are being under now screening process in the context of different solid tumor indication, ovarian, pancreas, and mesothelioma. And so this is give us a confidence, cautiously optimistic, say, okay, this is a target that is expressed on the surface that is selected specifically on cancer, highly specifically expressed on cancer versus the adjuvant normal tissues and so forth. So we went through then defining the Fab of that particular and constructing our CAR. This is an RBV third generation where we insert in our target. This is nothing new, but this specifically using the difference we're using gamma delta T cells. We have a proprietary technology that defining, isolating and expanded and engineered them in the context of our target, or AI-derived targets. So we are using this unique subpopulation of T cells, gamma delta T cells, so very unique, with a very nice safety profiling that is being studied in the stem cell product. Gamma delta, it's historically speaking, a very safe T cells and therapy. And we've been learning how to isolate them, expand them, and that is the key, and how to engineer them. So there is a little bit of trick. We spent a few years on defining this manufacturing system. Our in-house development of gamma delta T cell expansion product is in total of 2 weeks, which we can really expand our gamma delta to and engineer them with our AI-driven target technology. So these are some results of our in vivo efficacy of our CAR gamma delta T cells, which interestingly enough, we have done it with several different animal models, and from also testing, and not just in gamma delta, but also in alpha beta, with different, almost nine different models in solid tumors. This model is actually a modified NCI H226, which it's basically a lung and mesothelioma system. We allow to see that that when it's been treated after almost 15 days, you can see in less than 3 days a complete regression of the tumor on the group of anti-isomeso CAR gamma delta T cell versus the control, the cell control, and the gamma delta non-engineerized and transduced. So that was a quite in the beginning we looked at it and said that doesn't look real. We repeated so many times, and the results are the same, also independent data. So we were very pleased to see that if just half of it, this efficacy will work in our future phase 1 clinical trials, we will be really having a very effective and safe and efficacious therapy in solid tumor. So what you can see also on your left, that there is truly not loss of weight or general health on any side effects. And we have done preclinical PK and study that the FDA was requiring during our submissions. So that truly shows that all the groups have not, even treated, non-treated, tumor was not losing weight, so truly didn't have any issue. But these are the results of the different cytokines panel to check on this assay of the cytokine syndrome, and also any other unwanted storm of cytokines produced. You can see here in the panel that we are providing. Truly, it's the transduced and non-transduced was truly the panel was no significant difference, so truly not any side effects. We also did the pathology of these groups of mice several times required by the PK and PA for the patients for the IND that we have been submitted, and a different time on 72-hour cytokines. So truly, we didn't see any major differences. On top of it, on the pharmacokinetics, on the peak of the CAR gamma delta T cells in blood, after 3 days injections, the peak of T cell number correlated with the timing of the tumor burden began shrinking. So they basically disappear. And after 45 days, the expressions after the infusions, you can see that after 45 days of tissue distributions on blood, spleen, lymph nodes. So it's a truly pretty safe system. And you can see that gamma delta slowly disappear in the blood after 20, 25, on 45 days are the last, and so forth. So on the results of the CAR gamma delta PD, as you can see on the toxicity persistence in vivo, that's quite interesting on the tumor growth versus these are 3 groups that we have been testing. 2 groups have been testing on the CAR gamma delta treatment groups. You can see the persistence in vivo measured by the re-challenging the experiment on 45 days after the gamma delta infusion. And groups of 5 mice were injected with the NCI H226 cells, following the same protocol the first tumor injection. So the interesting part that the tumor volume and mice weight was measured, indicated above, while the blood was drawn for the measurement of the circulation CAR gamma delta T cells. And you can see that they were observed after 5 days a peak on the re-challenging, which truly it's quite interesting. There was a challenging. You can see that the tumor didn't grow in the survival CAR-T after re-challenge. And so we feel that there is a secondary memory on that, and also the disappearing of the CAR gamma delta, I would say, safely in the context of the 40 days, 45 days. So in conclusion, what I showed you here very briefly highlights, not to go into the details, even though we had a full-fledged poster presentations in AACR last few months ago when we presented this full study, and showed truly that we are able to generate an autologous and allogenic off-the-shelf gamma delta with an AI-driven targeted technology. That in vitro, in vivo, the data in vivo has been repeated several times in different models, lung, pancreatic, and ovarian cancer. Truly showing the efficacy of this therapy, and we are preparing, of course, working with FDA to address all the question of manufacturing. So in conclusion, we were able to show a target that we can generate targets specifically, showing some in vivo data, showing the cytokine profiling and the truly a low risk of CRS due to the uniqueness of the gamma delta, some in vivo data, showing the ability to complete the rejection secondary tumor injection with our AI iso-meso-CAR gamma delta. So with that, I would like to thank our team. This is a group of very talented scientists that joined our team in the last few years. Have been really taking the next level our product. Hopefully, we will be able to bring something that would be effective in this very hard challenge for all of us in solid tumor. So with that, I would like to thank everybody, and I'm open for any questions. Thank you again.
Thank you for that. It's an interesting pipeline, and it's near and dear to our group's antigen discovery efforts. The splicing work that you kind of highlighted. What is the mechanism of the splicing event? Is it just simply alternative splicing, or is it due to it doesn't look like there's a splice site mutation, or maybe that's something that you're building into it? What are your thoughts there, or observations? I'm actually kind of interested in the CTAs, because those are usually not mutated, but there's a lot of isoform usage.
Yeah, we had developed a software, a cancer splice system, which is able to define with the variation of splicing, not truly mutations. Yep. I cannot go into the detail because it's a proprietary technology at the moment. We are in the process of refining the patent. But you're asking a very important questions, which I will be able to address more in detail probably in the next meeting, which I will be able to allow to talk more in detail. It will be actually a talk on cancer splice and how the mechanism and how we defining, and truly it's a combination of technology with computer scientists, Dr. Ryan, ex-MD Anderson, and it's truly work on developing the last 5 years on this particular software that truly is a combination of talents, and I will be more than happy to present something in the next few minutes.
Yeah, I appreciate that. That's I know it's difficult in understanding the cancer specific splicing for certain genes is extremely important. That would be super cool. Now, is there any other questions in the chat here? Trying to look right. Let's see any other ones. I guess I can ask one follow-up then. So, you're identifying these. You have these rules of splicing, and you've developed this algorithm now. It's trained on short read NGS, or are you thinking about, or are you integrating some longer read technologies to either get cis and trans types of information?
It's a story, and as I was mentioning in the beginning, the data training started with the CTA. As you mentioned, I've been working on CTA for over 20 years. Yeah, we defined in the beginning 7 years ago, starting just the data setting on the CTA, some of the WISE1, trying to validate the targets, and then also some of the targets I discovered, like sperm protein 17 in the context of multiple myeloma and ovarian cancer. They went to the clinic, and they showed that that could potentially could be extremely valuable data, and not just that one, but there are plenty of targets from the Rosenberg teams, and so that in mind, definitely it's an omics comprehensive system that we are using in the context on NGS. but truly it's, as I mentioned in the beginning, it's a truly an omics comprehensive system that uses the cancer splice through re-epitope system, was a modification of the John Hopkins or Masato software. We have a suite of software that, and I will be very happy to provide more information on our platform. That is, it's in one simple page. You can truly get a full-fledged target that goes through the NGS, to the short frame, to the long frame, to even creating epitopes, because the refining on the surface, also there is also refining of the peptide, the short and polypeptides, which allow us to defining the anchor and the binding of truly what we want to define in the context of CAR. We have two different, we are not yet presenting also our library on T-cell receptor, on TCR, which we are working in combination with it on the TCR and CAR. But it's a comprehensive application. As you mentioned before, very difficult finding targets for the heterogeneity, very hard to creating a system that can hit some of them. There's a lot of false positive in the context of MHC class 1 for T-cell receptor and also for CAR. So truly, we are looking at, and that there is a lot of work behind the scenes with our computer scientists, our validation team, biomarker teams that is working on it. But to address your question, is an omics fully integrated system just to allow to define some of the target. We have 5 targets in the pipeline right now. They're going through the validation discovery, and believe me, it's as you know, it's probably one of the hardest aspect of our field.
I agree wholeheartedly. The splicing side, this is where we're starting to think it's very difficult to understand the heterogeneity and actually understand the you can't really understand the rules of it. You can get there. You can get there and then impute sometimes, what's going on. But it's we could talk for hours on it.
I think what you're doing is very, very sexy. Your approach is very sexy. I would like to talk more. Probably.
Yeah, I'd love. Yeah, we can catch up offline.
I think we have a lot in common. A lot of things that we can actually talk.
Yeah. So one final question. So very interesting work. Just curious, from PK study, CAR plus number decreased dramatically at day 20. Would you consider this might indicate potential issue of shorter persistence?
Oh yeah, that is actually a very good question. So we don't expect, this is a therapy, the persistence in a gamma delta is very well known. There is a very short, and when they talking about NK or gamma delta innate immunity for persistency and how long will last, 3 to 6 months, can we engineer them to be longer? We definitely have a different kind of approach. We will be able, we're truly looking at in a live drug that can be multiple doses, because it's an off-the-shelf. We can provide through our easy step manufacturing multiple doses and allow it to follow up. But there is a secondary memory, which of course the persistency is one of the issues that everybody knows, and I agree this is a very good question. Which, yes, we have something in development to increase that, but truly is an important aspect in the context of what we are trying to achieve in the process of having an off-the-shelf allogenic product that can be cost-effective, available, and eliminating. Think about it as a surgery through cell therapy. It's a chemo that is being given with a high quality life expectancy. If I just seen, I'm optimistically cautious here. If I see just 25% of what I see in our animal models effective in a human in our phase 1, I think we will really help our field and our patients. Persistence is important in autologous, but definitely it's important for everything. But if it does the job, and then we can, with the secondary memory, which is being released, and we show that with the re-challenge. There was an important slide. This is the reason why we put it in the deck, and I would like to emphasize. I think we can actually create a treatment that can be given as needed, and patients can live with minimum residual dose of cancer, and we can treat it and work with that. We don't expect to have a magic bullet. It's a modular therapy. It's a combination of therapy, and sometimes can be also a short therapy that can really help. And that is our philosophy.
Thank you so much.